Professor, good morning. Can you look at the chat, please?
I'm sorry?
Can you look at in the chat, please?
No, I will not do that. Can't do that.
Okay.
Okay. Good morning, everybody. Just a brief word. I thought everybody did quite well last week on the exam. Hold on a second. Everybody did quite well last week, and so nice job. Any questions regarding that? And if not, we'll put it to bed and move forward. Any questions regarding the exam? Okay. So that's all.
Yeah, I have one question.
Yeah.
I have one question. Can I ask?
Yeah, please go ahead.
Yeah, I would like to know how muscle tissue is considered as a epithelial tissue, please. I would like to
Not
understand.
considered epithelial tissue. It's considered tissue.
Sorry.
I think he meant that, and question number, I think 33.
Really, I don't know the question numbers.
He meant that epithelial tissue is connective tissue. He meant how is that possible? How is epithelial tissue connective tissue?
It's,
connective tissue. Yeah.
It's considered to be connective tissue because it's on the lining of all organs and internal organs. We talk about the connective tissue. Folks, if you have a teacher that tells you something different, I test you on the notes in my exam. I'm not going to compete with other teachers, argue with other teachers or professors regarding that. If you read the materials, you should do well in the exam. You all did well. Then argue for 3 points. Really don't bother. If you had a question regarding any other content, but you're going to argue for points, this is not the place or time for that. I answered you as well in email. Any other questions? Okay. We're going to move on. The next exam will cover everything from this point forward. So we'll talk about muscle physiology today. We'll go through the slides, and at the end there'll be a couple of videos that you can look at your own leisure that will provide a review of the material that I covered today. I won't be with you for the those videos, but you can look at that on your own time. So a lot of the slides I'm going to have will have some Hebrew to help you out regarding that. There's 3 different muscles that we're going to discuss. We're going to discuss cardiac tissue, smooth muscle tissue, and skeletal muscle tissue. All 3 of them have distinct characteristic differences that provide a specific role in how these muscles will operate. The cardiac tissue is interesting in that it gives a very similar striated appearance as you'll eventually see in skeletal muscle. And then it's surrounded by the connective tissue and has a healthy blood supply to it, which are called the coronary vessels. We'll talk about that at a later time. The epicardium is the connective tissue on the outside. The importance of the prefixes will help you understand what the role of that particular element is. Epi, the prefix epi means outer layer, versus endo meaning inner layer, and myo is just muscle cells. So it's pretty easy to understand what the role of this particular tissue is based upon its name. So endocardium would be the inner lining of the cardiac muscle. Myocardium is actual muscle. Myo is going to tell you that. And the epicardium is the outer layer of the connective tissue layer of the muscle or the heart. You'll see here coronary arteries, and what's important to understand is that the coronary circulation is separate from the systemic circulation going to the rest of the body. So it bifurcates from normal arteries, and it has its own blood supply. When this coronary artery becomes blocked, and we'll talk more about that later on next week, next 2 weeks, when this coronary artery comes blocked, there's no blood flow and hence no oxygen supply to the muscle. If there's no oxygen supply, then the tissue around this area becomes ischemic and eventually will die. Without oxygen, the tissue will eventually not be provided nutrients and will eventually become necrotic and no longer operate. This is what's termed a heart attack. A heart attack occurs because the tissue surrounding the heart no longer is functional, and it no longer competes, no longer responds to any electrical signal. And if you have 5 coronary vessels surrounding the heart, and if there's certain parts of the heart can actually stop working, become ischemic, necrotic, and die off, but the heart can still be functioning. So it depends a lot about the extent of the ischemia within the heart. The sac around the heart is known as the pericardium. Actually, the coronavirus, where the concern is for young individuals, where most of the knowledge you get from the media is that the population that's most risk for dying from being inflicted with or catching the COVID-19 virus are older adults or individuals who have a suppressed immune system. Well, actually, there's some interesting data that young people who catch the disease, they may not be at risk of dying, although some have, but the biggest problem that we see with people with COVID-19, young people, especially competitive athletes, they get something called pericarditis, which is an inflammation of this connective tissue that surrounds the heart. And this could be lethal, eventually will not allow the athlete to any longer participate in competitive activity. So it's a delicate or inflammation of the sac that's surrounding the heart. It's quite dangerous. Okay? So this is a bit about the anatomy and physiology of the cardiac tissue. If you look closely at cardiac muscle, you see it provides that striated appearance. So it provides the contraction, which we'll talk about in detail, muscle contraction. It has this, but what's unique about the cardiac tissue is it contracts in a very organized manner from the top of the heart to the bottom of the heart, from the atria to the bottom. And we'll speak more about that next week when we talk about cardiovascular physiology. But as you can see, with the cardiac tissues, there's several different layers that we've talked about. And this is some of the endothelial tissue that we see here underneath the smooth endothelial tissue, which lines the inside of the heart. This is the endocardium that we talked about, the bottom layer, the epicardium, or dense fibrous layer on the top of the heart. There's a loose connective tissue underneath it, a layer of mesothelium, another connective tissue, separating the epicardium from the cardiac myocardiocytes. This area of all these connective tissues is the parietal pericardium, and then you have the cardiac tissue, known as the myocardium. You see here in the middle. So the cardiac tissue.
Professor, what is pericardium again?
The cardiac, because it's cardiac tissue. Okay. So.
סליחה, פרופסור, מה הקיצור של פרי? כמו שאמרת שאנדו זה...
Endo's internal, inside.
אז מה זה פרי? כאילו בדרך כלל?
Peri is the whole thing, covers the whole area.
Or.
So the cardiac muscle is very similar in structure and the mechanism of contraction of skeletal tissue. And one of the things that is separate, different in the structure of the cardiac tissue is it has intercalated discs, as you can see here within the muscle. These intercalated discs are unique formations, structural formations, that are found between the myocardial cells of the heart, and their primary role is enhancing the bonding between cardiac muscle cells and transmitting the signals between the cells. And why this becomes important is that the heart is going to contract from the atria, the sinoatrial node, through the atrioventricular node, through the rest of the myocardium. So it contracts in a specific way in an organized fashion that allows the heart to pump from one chamber to the other chamber, allows you to continue to breathe and function normally. So the intercalated discs play a very important role in transmitting the signals between the cardiac cells. And that's the unique thing that differs between muscle, skeletal muscle, and cardiac muscle. The skeletal muscle will contract entirely at one time. The cardiac muscle will contract in stages.
Professor?
Yes.
What exactly is there in the disc? Is it just like a suture kind of thing?
Yeah, it's just a simple, it's a connector. It's a connection, that's all it is. And I couldn't get into the molecular makeup of these discs. There's proteins within those discs that provide this integrity to the area to hold it together.
Professor, do we only see cardiac tissue muscle in the heart, or do we also see it, for example, the muscles in the arteries? Are they also cardiac muscle?
No. The only cardiac tissue you're going to see is in the heart. Only in the heart. We'll talk about that in a moment. It's a good question. In the vessels is a smooth muscle. And once you understand what the responsibility, how things flow, you'll get a better understanding of why that is. So in the heart, next to the intercalated discs, there's gap junctions, as you can see here. Here's an intercalated disc, and next to it is a gap junction, next to another cell with intercalated disc. These allow for the action potentials, the electrical signal that we talked about, to spread from one myocyte to the next. So how the muscles will contract will from one cell to the next cell in an organized fashion, and it's the electrical signal is going to be passed through these gap junctions. So we'll talk more in depth about cardiac tissue over the next couple of weeks. So we'll stay focused on muscle, and now move on to shirim chalakim, or smooth muscles. And as the name applies, there's no striated appearance. It's actually a smooth appearance. And this is all involuntary. You can't say, I'm going to contract my intestines or my vena cava or aorta right now. You don't have the ability to contract your heart. You don't have the ability to. Well, the other thing about the heart, it's also it's autonomic nervous system. It's also important to understand. The heart is innervated by the autonomic nervous system, as is smooth muscle. So these are completely involuntary. You don't have the ability to control heart rate. You don't have the ability to control contraction of smooth muscle. So together, the smooth muscle with the cardiac muscle makes up about 10% of one's body mass. And these appearances are quite smooth. There's a picture of smooth muscle. Here you can see smooth muscles are found in internal organs, including blood vessels, the lining of the stomach, or of the esophagus or trachea. These are all examples of where you're going to find smooth muscle. And the thickness of it's going to change as you move down. We'll speak more about this when we're talking about vasculature. As we talked about smooth muscles, and again, it's several other pictures of it, is controlled by the autonomic nervous system or autonomic motor system. And within the smooth muscles, there are various types of epithelial tissue underneath it. But the primary role of the smooth muscle is to provide stalactitic pushing the materials that are within the lumen of these organs forward. So as food is being digested, pushed into the stomach, and goes through the esophagus, into the stomach, through the small intestines and the large intestines, is the contractions of the smooth muscle allows it, peristaltic movement allows it to move from one, quite slowly, move from one section to the other where digestion occurs. So it's not as if there's a contraction of the entire stomach or the small intestines, as you see in the regular muscle. It allows for a smooth flow from one end of the digestive tract to the other end of the digestive tract, allowing digestion to occur. So if you think about the development of the muscles and the system, how it's being operated, it makes a lot of sense that the smooth muscle would contract in a way that allows peristalsis of movement in a specific direction. And as you can see, the smooth muscle, there's an elaborate neural network that allows it to contract in that manner, as well as various layers of connective tissue and smooth muscle tissue that's all that we see surrounding every organ.
I have a question.
Yeah.
Can you go back? Can you go back to the previous slide? Okay, here. When we say lining of stomach or intestines or something, which part of this is it referring to? The next one.
The entire organ, the internal, if you can see here, you have the epithelial cells that are inside here, the connective tissue.
Mm-hmm.
Then you have an outer lining, again, a connective tissue, and then between the two, you have the actual muscle. And this is what's causing the movement of in this example, the stomach, of the food particles from one end of the stomach to the other into the small intestines. So as we talk about digestion next semester, we'll get into much greater detail of what happens in those particular organs and how the movement actually occurs. But for now, you understand that the smooth muscle doesn't have the stride appearance, so you don't have what's known as the sliding filament theory. And we'll talk about that in a moment. What you're having is a peristaltic contraction of begins from one end of the muscle, contracts to the other end of the muscle. It's similar in a way we talk about cardiac tissue. And understanding the physiology of how the heart works, the physiology of how food is digested and the importance of placed in these organs, provides a better understanding of why these muscles contract the way they do. It makes a lot of sense physiologically in the role that it has. And the body is built with logic. And one thing that's beautiful about science is very logical. And if you understand what the physiological role of that particular organ is, or what that particular element is supposed to do, you have a pretty good idea of how it's supposed to operate. That being said, when there's a problem there, you also have an idea of where you can look for that problem. It's no different than, say, a mechanic looking at your car. He knows that every piece of equipment in that car is supposed to do, your engine, your carburetor, radiator. So he knows how everything operates in a very logical order, and when it doesn't operate, he can go and fine-tune what's going on. No different than physiology, is that we understand what every organ is supposed to do. The question then becomes is, once you understand the logic behind it, it's very easy to understand what is going on. So as we move forward into the smooth muscle, we've finished that. And most of the lecture today is going to go into skeletal muscle. So we'll come back more to cardiac and smooth muscle when we start speaking about those organs. For example, the next 2 weeks we'll be talking about cardiology, or the cardiac physiology, and we'll have much greater time we'll spend on the heart. And then next semester we'll talk about digestion and physiology of digestion. We'll talk more about the smooth muscle as relates to that part of the body, and then the vasculature we'll speak about that as well. So the majority and the remaining of today's lecture, we're going to focus on skeletal muscle, and there's about 650 skeletal muscles in the body, and they make up about 40% of the body mass. Now, in contrast to the smooth and cardiac tissue, skeletal muscle is voluntary, meaning that we can control whether we contract it or not. It doesn't control us. We control whether we move. We control whether contraction, whether we want to fix something up, and we determine how much force we're going to apply to them. They're also known as striated tissue, and the striated tissue becomes because of specific proteins within the muscle that we'll talk about. The benefit of skeletal muscle, it can contract quite quickly, and we can have a gradations of force from something very light to something that's maximal. We have the ability to control that. The skeletal muscle has a number of functions in the body. Obviously, the most important one is movement, whether it be locomotion or movement of the upper body, movement of the neck, breathing, respiration. These are all important components that we utilize. Body posture is also quite important, and it provides a protection for internal organs. Actually, with certain athletes involved in contact sports, increasing muscle size becomes a very important aspect of preparation to try to protect your internal organs from the contact of a competitive game. It also is able to produce heat. This is important, especially in the cold, the shivering response of muscles to be able to generate heat. So as the muscles become active, heat is generated. That's what causes elevations in core temperature. And also, it will help restore venous blood flow as we contract muscles in the lower body and the abdomen. It brings blood back to the heart. This is especially important if you serve in the military and you're performing guard duty, having to stand in the heat. Blood had a tendency to pool in your lower body. Some soldiers who were standing upright for long periods of time may have an episode of syncope, which means fainting. And in order to avoid that, I'm sure you were told by your commanders is to make sure you contract your calves as you're standing at attention. The contraction of your calf muscles, will have blood flow come back to the upper part of your body. It's a very effective way of making sure that blood flow continues. If you stand at one spot for too long a period of time, especially in the heat, the blood will pool and you have a chance for fainting. So by contracting the lower body, you'll have a chance to have return flow of blood to the heart. And finally, the muscles will help absorb shocks resulting from body movements. A very important point of that as well.
What is the last one again?
It helps absorb shock.
What does it mean? Thank you.
Okay, so this is a picture of a muscle, and it's important to keep in mind here, if you know what a babushka doll is, those Russian dolls, where you take out one doll and the same doll underneath, take it out, the same doll underneath that, just smaller and smaller, and you keep on going to the end. Muscle is very similar to that. We have the entire muscle, as you can see here, and it's surrounded by connective tissue, the epimysium, as we talked about. You have the endomysium underneath that. You have within there, you have the muscles. Now, as you can pull out, there's bundles of these muscles, and the bundles within the muscle, the whole entire muscle, is known as a fascicle. Each bundle is known as a fascicle. So that's this entire bundle here is known as a fascicle. And with each fascicle, you have other bundles, and these are known as the muscle fibers. So in each fascicle is comprised of muscle fibers. Okay. And you have connective tissue that surrounds each of these, and these are known as the perimysium. The connective tissue surrounding these fascicles. Around the fascicles, you can see that it's vascularized, meaning there's blood vessels here, clay down. There's also the innervations, the axons, the motor units, motor neurons that are here. And then we see them on the muscle fibers as well. And we talked last week about motor units. So motor unit is the actual nerve and the fibers that it innervates. When they contract, it's an all-in-on principle that we'll talk about that later. So surrounding the muscle fibers within there's even other strands, other tissues, which are known as the myofibrils. And the myofibrils are comprised of two myofilaments. Okay? The myofilaments contain two proteins known as actin and myosin. They are known as the myofilaments. Okay? So again, as you take a look at this entire muscle, it goes from one bundle to then a smaller group, to a smaller group, to a smaller group. The myofilaments are not the last part of it. They comprise the smallest functional unit of the muscle is known as the sarcomere. We're going to talk about that as we move forward.
Professor, I didn't understand. What's the fascicle exactly?
The fascicle is a bundle of muscle. The muscle is comprised of various bundles. The fascicle is one bundle.
Isn't that the perimysium?
No, the perimysium is the connective tissue that surrounds it.
Perimysium surrounds the fascicle?
Correct. And so you have in the muscle, you have various fasciculi. So you have one fascicle, many fasciculi.
Sorry, Professor. So you say the filaments, each one includes actin and myosin?
The myofilaments are actin and myosin.
So it's the last part of it.
Those are the myofilaments collectively. The actin and myosin proteins are known as the myofilaments.
Right.
That's what gives the muscle its striated appearance. Actin is considered to be the thin filament, and myosin is considered to be the thick filament. We have many slides depicting that. So a muscle connecting the muscle to the bone. So a muscle is attached to a bone by a tendon. And the role of the tendons is to transfer the tension created in the muscle to the bones. This way it eases the tension from the muscle. Tendons do not have the ability to be stretched. If you stretch it too much, you actually can get injured. And we'll talk about sensory receptors within the tendons that actually measure the stretch. That eventually keeps you guys very busy as physical therapists, especially if you become sports physical therapists. So it will transfer any small tension develops in muscle to the skeletal system. So it has a very important role, and the primary, when we talk about a tendon or a guide, is that it connects the muscle to the bone, and it provides a measure of the tension within the muscle that's being generated that allows it to bring it to the skeletal system, offload some of that tension. So if we take a look at the structure of a single muscle, it's an interesting picture, and it's important that you really understand this. So we're now looking at a muscle fiber. Okay? And within the fibers, as we talked about, we have the myofilaments, the myofibrils. Within the myofibrils, we have the myofilaments, as you can see here. So each of these dots here, you see there's thick dots and thin dots. Does everybody see that? Okay, you all see that now, right? That's actually the myofilaments. And the thick dot you see here, the large circle, that's known as myosin, the thick filament. The small dot here is known as actin, the thin filament. If you continue all the way through, think of these as myofilaments that go from one end to the muscle to the other end of the muscle. That's what gives this striated appearance, as you see here. Thick and thin filaments, it gives it its striated appearance. So it has really importance of why the muscle looks the way it does, and it's simply related to these myofilaments. Okay? Now, we talked about the myofilaments is not the end. It's not the smallest functional unit. The smallest functional unit is known as the sarcomere. And you're looking at the sarcomere, it's from this point here to this point here. That's considered to be a sarcomere. That's the smallest functional unit within the muscle. Sarcomere. So at the end, the demarcation for each sarcomere is the Z line here. Beginning and end is the Z line. Within this, you see these thicker lines and thinner lines. Those are the thick and thin myofilaments, or the myosin and actin proteins. Now, within each of these myofibers, and keep in mind these fibers, we talked about going to innervate. The motor unit is the neuron and the fibers that it innervates, not the myofilaments, but the fibers. So it's right here. This is the entire fiber. So what is important for you to realize is in this fiber, you have an elaborate network, a tubular structure known as the transverse tubule system. This has a very important role, and what's important to realize is that within this cell, you have these openings. And this is where the electrical signal will come through. If the action potential occurs, goes down the transverse tubules, where it gets to the storage site known as the sarcoplasmic reticulum. This sarcoplasmic reticulum is the maxan, the sidan. Or calcium. There's the storage site for calcium in the sarcoplasmic reticulum. And as we continue to talk about muscle contraction, you'll realize how important a role this is. On this picture of taking a look at the single muscle fiber, so you see the transverse tubule system, the sarcoplasmic reticulum, which provides a storage site for calcium, provides a pool of calcium, but you have within the fiber, you have the mitochondria. This is where ATP is partly produced, right? It's produced in large quantities through oxidative phosphorylation. We have a nucleus. And then we have within there the sarcoplasm. Within between the fibers. We'll talk more about these areas within the sarcomere in subsequent slides. But this is important that you are able to identify everything on this picture and understand their physiological role.
Can I ask a question?
Sure.
Will electrical charge in the muscle fiber always appear as calcium?
If there's an electrical charge that allows muscle to contract, calcium will always be released. And you'll find its role in a moment, what the precise role that calcium has.
What are the transverse tubules? What do they do?
The transverse tubules is a sort of like is a network of tubes, like a plumber, like a plumbing system in your muscle, that the electrical signal goes down. So if there's an action potential that's sufficient, that electrical signal that goes from the nerve to the muscle travels through the transverse tubules into the sarcoplasmic reticulum. Is that stimulus that's going to cause a release of calcium from the sarcoplasmic reticulum.
And it wraps around all the myofibrils?
It goes through the myofibrils. It goes through the myofibrils. That's correct. So keep in mind the motor unit is the neuron and the fibers that it innervates. So if we were to draw the motor neuron, let's see a picture of it later. Okay. We have the acetylcholine vessels, and on the surface here you have the postsynaptic membrane. This is synaptic cleft, right? There's a stimulus that comes down, and from this direction, sorry about that, and releasing acetylcholine interacts with its receptor, an action potential occurs, and the electrical signal goes down the transverse tubules into the sarcoplasmic reticulum, releasing calcium.
Professor, so one single muscle fiber is actually a single cell?
And a single cell, yes, in certain disease states, of course it could. There's a number of reasons why a signal could fail. I was involved in something last week. We were talking about a very interesting situation that happens in a small amount of the population, but it's very serious. It happens in young folks especially. It's Guillain-Barré syndrome. And it's a situation in which there's a degradation of the myelin sheaths on nerve endings, in which the nerve impulse no longer gets to the muscle, and it leads to actual paralysis in the lower body. And so there was a situation that came from a virus. And what was interesting, the situation that happened, there was a potential a virus, and one of the individuals actually no longer could feel their lower body. And so it was a very high risk for that sort of disease, which the person needs to be get because if it continues without getting early treatment, it can lead to paralysis. So there are certain types of diseases, viruses, that can destroy this. And we'll talk more about that. Chemical attacks, for example, they're designed to eliminate acetylcholine. And if you eliminate the acetylcholine as a neurotransmitter, you'll be unable to contract muscle as well. We'll talk more about that as we move forward.
פרופסור,
Yes.
אז האקטין והמיוזין זה בעצם ביחד הם יוצרים סרקומר?
נכון. ועוד קטנים, אבל זה הם המיופילמנטים.
Okay.
Professor, what part does the calcium play? I didn't really understand that.
Yeah, we're going to go over that about many times, but I didn't mention it. So we're going to talk about it as we move forward. I haven't mentioned the role of calcium. I just told you it plays a very important role for contraction. You need to understand right now where it comes from, and then as we talk next few minutes, you'll understand the role of calcium in muscle contraction.
Also, I didn't understand if the motor neurons will contract a sarcomere or a whole fiber.
Okay, good question. I want you to understand it. It's going to contract the smallest functional unit. Okay, זה יחידה הכי קטנה פונקציונלית הוא sarcomere. For the fiber to contract, it's contracting all of the sarcomeres within that fiber. But where the contraction occurs is within the sarcomere. קיבוץ קורה בתוך הסarcomere, או קורה כל הסarcomere בתוך הסיב מכווץ באותו זמן.
Okay, so when there is
תחת הציב. בתוך הציב יש את המאופילמנטס ויש את הסarcomere. הסarcomere we talked about זה ה-functional unit, יחידה הפונקציונלית הכי קטן שיש, כל העבודה קורה בתוך הסarcomere. Okay?
Yes, but we also learned that it's like all or nothing when there is an action potential. So I'm trying to understand if it's one sarcomer that will contract or it will contract the whole fiber. That's what I'm trying to understand.
All the entire motor unit, when it's activated, will contract. That's the all-in-none principle. That's correct. What's contracting is the sarcomere of that entire fiber. Okay? And you'll see, you're gonna get sick of it by the end of the day. We're going to talk about it by then. Okay, so sorry. Okay, so muscle cells are found inside the connective tissue in the fascicle bundle called the sarcolemma. Okay, surrounding that, and the fasciculi together are wrapped in the muscle envelope. Okay? And so you have a picture here in Hebrew to help you with this. Just a bit of a review. We talked about this, the structure of the muscle. Again, here's the gid, etzem, shrir, fasciculi, called the agudat shrir, bivrit, כלי דם, ציב שריר, בתוך הציב יש את המיוקרדיטים, דגימות חיבור.
פרופסור, ציב שריר, או זה תא אחד, נכון?
No, יש many תאים בתוך sarcomeres.
Okay.
So the myofibrils, as we talked about, again, they are made up of sarcomeres, right? So the myofibrils that come out of the sieve are made up of sarcomeres, and the sarcomeres, the smallest functional unit of the myofibrils, are made up of protein filaments that's known as myosin and actin. And it's the interaction between these filaments that is responsible for muscle contraction. You need to understand that. That's important for you to understand that it's the proteins within the muscle. It's the actin and myosin proteins that are known as the filaments that are responsible for muscle contraction. Myosin, which is considered to be the thick filament, is composed of two protein strands that are folded together that forms a globular head at one end. The thin filament is considered to be actin. It's called actin, and it is two other proteins that are involved with it. It's called tropomyosin and troponin. One end of the thin filament is attached to the Z disc. And we talked about the Z discs line the sarcomere, or the beginning and end of a sarcomere. And embedded in that Z disc is the actin. In the middle is the thick filament, or the myosin filament, that has the globular head. So here we take a look at another picture of a single muscle fiber. Here we talked about the myofibrils are consisting of two smaller units called myofilaments, actin and myosin. Here's the striated appearance of muscle. And this is how we look at the Z line. And within one sarcomere unit here, okay, so one sarcomere unit, Z line to Z line. Here's the Z line right here. See one Z line here, another Z line here. There's various different zones. And this is so you can see the striated appearance. In the middle of one sarcomere, this is one sarcomere. Embedded here is the actin. See the myosin, and in the middle, in this space right here, this is the M line. On both sides of the Z line, as contraction occurs, it moves in these two directions, shortening. And as you begin contracting the muscle fiber, you see a shortening of the H zone. This is considered to be the H zone. This area here shortens, and what is happening is these fibers begin to close the distance. They're sliding over one another. Hence the term sliding filament theory. It was developed by Huxley in the 1920s, I believe. But it's quite interesting how this happens, and the evidence is pretty strong. This exactly is what's going on.
אז רק כדי להבין שהבנתי, אז האקטין מחליק על המיוזין.
הפוך. Because actin is embedded in the Z, right? It's embedded at the myosin. So the myosin head, and we're going to see this again, attached to the actin molecule, ratchets it, and moves it this way. As it moves it this way, you actually have a closure of the H zone. Closure. The M line is moving closer together, both sides, and you have a muscles, the sarcomeres become shortened. Hence the contraction. So the entire fiber is happening in all the sarcomeres.
How is the actin connected to the Z-line?
The actin is connected to the Z-line through proteins known as desmosomes. Provides, maintains the integrity. Actually, when we exercise with high intensity resistance exercise, we create damage within the muscle fibers. It's natural, and it generally see what's known as something called Z-line streaming, where the integrity of the Z-line is broken, is reduced. It's during this recovery process that we start to see an improvement in muscle size and muscle strength. So the saying that you may have heard at one point in your life, no pain, no gain, there's actually some truth to that. Because in order to actually build muscle, you have to create micro damage. Micro damage in the muscle actually results in a breaking down, degradation of sarcomeres, and over time, the ability to break it down, you become more resilient. And that adaptation or resiliency is developed over time through training. If you've ever done something for the first time, lifted weights for the first time, you remember how sore you were after you did that, and that soreness peaked 48 hours after. We'll talk more about this when you take exercise physiology, but that soreness peaked 48 hours, and that just resembled the damage that was occurring within the muscle. And the next time you worked out from the same exercise with the same load, it felt much better, and to the point where you were able to function after every workout. Now, for competitive athletes that are looking to build lean body mass and get stronger, they need to push themselves every workout, try to create that sort of stimulus that creates an adaptation within the tissue. So if you continue to train at the same intensity, relative intensity, or same absolute intensity or load, eventually it's going to become very easy. The body adapts, but it's no longer providing the same stimulus, so your ability to increase strength becomes reduced. And we'll go over that in much more detail when we take exercise physiology for you guys. It's next year. But it's very, very interesting to study muscle physiology. And it's very important in your careers as physical therapists as you begin to rehabilitate individuals, whether they're 80-year-old grandfathers and grandmothers, 20-year-old athletes who are recovering from injury. The adaptation process is the same. What loads you use may be different depending on the ability of the individual, but the idea behind adaptation is critical to the work that you do. This is an electron micrograph of the myofibrils, and we can see here is fatty deposits amongst these are we put nucleonim in here. You can see the different colors of the antibodies. You can see different immunofluorescence. You can see the colors of the striated fields. It's pretty neat. Okay, so we'll talk now about more about.
Professor, לא הבנתי מה ראינו בתמונה הקודמת.
I mean, you don't have experience taking a look at high electron microscopy pictures? Really? Where have you been? What you're looking at is a fiber, and it's, as I mentioned, it was stained and allows you to really see it nicely the striated appearance of the fiber. That's all this is. So what's really neat about this particular picture, again, you're looking at an electron microscope, the most powerful microscope there is. You can see the actual Z lines, you can see the sarcomeres within the fibers. That's what makes this really neat. And we'll show pictures towards, well, I'm not sure if I have those pictures, but this physiology. This gets disrupted after exercise. So we'll talk now about the sarcomere. Again, as I said, we're going to be speaking about the muscle, the striated muscle at nauseam and how it contracts. So the sarcomere is the smallest functional unit of the muscle. It contains the contractile elements that surround that each Z disc is the beginning and end of the sarcomere. There's an I band, which is a light zone, an A band, which is a dark zone, and we'll go over it if you don't remember it. The H zone we did talk about, that's the middle of the A band, right, where the muscle, where we see it begins to get smaller as we contract the muscle, as the fibers cross over or slide about one another. The M line is the middle of that H zone. Then you have the rest of the A band, and you have the second I band. Now again, we'll talk about that in more pictures, and here we go. So again, this is a picture of the fiber, and looking at a myofilament in terms of the sarcomere. And here we go. This is the thin filament, the actin molecule embedded in the Z line. We have the myosin thick filament, as you see here, with its globular heads. It's a little bigger that you can see here. Again, this looks.
What is globular head? You say
it's a protein, the globular protein. It's just the name of how the two strands of proteins, the myosin proteins, form this globular head. And this has a really important role that we'll talk about. So here's the actin molecule. Within the actin molecule, you see there's two additional proteins we talked about that are embedded in the actin. The actin is the backbone of this thin filament. Then you have the tropomyosin, which is a string-like protein that goes around the entire actin molecule. And at several parts on this tropomyosin, there's another protein known as troponin. So there's three proteins that are on the actin molecule: actin itself, tropomyosin, and troponin. Each of these have an extremely important role in muscle contraction. On the myosin, it has two strands of proteins that are intertwined that form two globular heads. We'll talk about, and these also have an extremely important role. And this is the functional part of the myosin molecule that actually interacts with the actin protein. And it's that interaction between the myosin head and the actin molecule which causes muscle contraction. So now we take a look at the area of the sarcomere. We've talked about the M line, the center line between the two sides in the middle, and the H zone is the M line is the center of the sarcomere. The H zone is the area that does not have the myosin that will contract. You have the entire A zone, A band. It's from the myosin one end to myosin the other end. And you have the I band, which is the clear part by the Z line on both sides. Okay, so now you're familiar with all the bands as well as the lines. There's another protein that's important. It's called titin. It's a relatively new discovery, but titin is also important regarding maintaining the myofilament integrity within another protein known as nebulin. So there's some people that do make a living doing work on research on muscle physiology, and they're looking at various proteins that maintain the integrity of the sarcomere.
פרופסור?
Yes.
יכול להיות שמישהו כבר שאל את השאלה הזאת, אבל רק בשביל להבין, המיוזין מתכווץ, ואז כאילו ואז האקטין מחליק מעליו?
Yeah, it's מחליק אחד מעל השני. We really haven't talked about לא דיברנו על קיבוץ, עוד לא דיברנו על קיבוץ שריר.
אוקיי.
We just talked about דיברנו על החלבונים בתוך הסוכנים, let's see. We're going to progress into that now.
Professor, what do we need to know about the theta and titin?
You just need to know that there are proteins that are involved in maintaining the integrity of the supplement. Okay. There's another picture of the muscle fiber and the supplement, and it gives you some information in Hebrew that you can use to This is another picture of electron microscope looking at the supplement, and more of a pictorial display of what this is with the myosin and actin myofilaments. See the globular heads. This would be the H zone where contraction will occur. Amylin would be here. This would be the I band. This would be E band. And again, some more things in Hebrew that allows you to study. Okay, another picture of the supplement.
Professor, can we have a break?
Yeah, I want to get to a certain point, and then we're going to make the break, okay? We will do it in about a minute or two. I just want to get to a certain area of the lecture. So this is, we talked about the sarcoplasmic reticulum and transverse tubules. We've already spoken about this. The electrical potential, the electrical signal will come through on the fiber. We're looking at the fiber here with the myofibrils underneath it. Inside here, the myofilaments. The electrical signal from the nerve comes through the transverse tubule into the sarcoplasmic reticulum, and this is where the calcium is released. It's really important that you understand this as we will move on to what muscle contraction. Okay? And we'll stop here, and we'll talk about the alpha motor neurons, and the muscle contraction will take a 15-minute break. Let's come back at 10:15. Okay, 10:15 we'll be in lecture again.